This method allows to investigate how mitochondrial redox state is affected in pathophysiological conditions. It can also be used to assess the efficacy of treatment strategies that aim at protecting mitochondria.
The main advantage of this technique is that it allows an organelle-specific assessment of dynamic and tracing changes of mitochondrial redox state in real time. This method can be combined with additional indicators to simultaneously record, for example, mitochondrial membrane potential or calcium concentrations in addition to redox state. This protocol can be applied to culture cells, tissue explants, and slice cultures.
Begin by optimizing the scanning confocal microscope settings. To do so, set the detector to 12 bits or 16 bits, activate the sequential scan mode, and add a second sequence/track. For both channels, select a pseudo-color lookup table that indicates over and underexposed pixels, and then select an objective suitable for the object of interest. Mount a cover slip with cells into the imaging chamber, add 1 ml of imaging buffer, and place the chamber on the microscope. Use the eyepiece and transmitted light to focus the cells. Record images with different pixel formats and pinhole sizes. Then record images with different laser intensities and accordingly adjust the detector gain and threshold. Finally, record images with different scan speeds and different numbers of frame averages.